Hey guys, I know I haven't really done one of these videos in a while. That is a video on pharmacology um, and substances. But I had a friend request that I do a video on alcohol. Um, Alcohol isn't the drug, and how it affects the brain and the body. And I thought it was a good idea. Kind of on a related note, I haven't really been making these types of videos as much because they require a lot more effort. Honestly, I mean, like I take preliminary notes before I make a video like this, even if it's something I know like fairly. If it's something I understand very well, I still brush up. I do research, I take notes, and none of this is scripted. But like I keep notes on hand and try and make everything as accurate as possible. But on the other hand, like if I rewatch a video and like I notice that something's inaccurate, it kind of it really bothers me. And in this type of in this subject, there's a lot of factors. There's a lot of things you can get wrong or misexplain, and so it's a little daunting sometimes because I want to get this like as accurate and comprehensible as possible. Um, and just the editing itself. I have to edit like an hour-long video, but whatever. It's like that's part of the fun. But sometimes I'm just not up to it. But I want to get back into it more. And alcohol, I think, is a good place to start. It's so pervasive in society. It's one of the most commonly used drugs. So yeah, I'm gonna first go over the kinetics of alcohol. That is how the body processes it, because I think there's a lot of interesting stuff there. And like in my video on DXM, I feel like it's necessary to explain the kinetics before the dynamics. And in general, it's like if something has active metabolites, you kind of have to get into that before you get into how it works on the brain, because the metabolites often play a role. And yeah, so I'm gonna talk about the kinetics, a little bit of the chemistry of different alcohols and how they affect the body and brain as well, because I think it's really interesting. And then we're just gonna get into the dynamics and a little bit on the mechanisms of addiction, and that should be about it. So yeah, I'll get right into it. All right, so first of all, lighting situation is not ideal. One of my lights went out and I have yet to replace it, so hopefully you guys will be able to see everything here. So I'm going to get right into the kinetics of alcohol. Alcohol is also known as ethanol, if you could see that, where it is a hydroxyl group, as in an oxygen bound to a hydrogen, and the oxygen is bound to a carbon, which is then bound to another carbon. So, and the ethyl prefix denotes that there are two carbons. Another alcohol like isopropanol, or let's just say propanol, prop would indicate three carbons, 1, 2, 3. So ethanol, it has a very high oral bioavailability, as in it's readily absorbed orally, about 80%. It's well absorbed, rapidly absorbed, and it's rapidly distributed to the liver, the kidneys, and the brain. It takes more time for it to reach the bones and other peripheral targets, but it crosses the blood-brain barrier through a process called passive diffusion, and it easily does so. It's a very small molecule, and small molecules like that tend to easily pass through the blood-brain barrier, which is the barrier that prevents certain molecules from entering into the brain from the blood supply. Let's just get right into the metabolism of alcohol. So the primary metabolic protein or enzyme involved in alcohol metabolism is called alcohol dehydrogenase, or ADH. So we got ethanol here, and it's metabolized first by ADH, alcohol dehydrogenase, and that removes the hydrogen here and forms acetaldehyde. And I'm just going to illustrate these so you can get a better picture of this, I guess. So after that, let me just say, acetaldehyde. After that, it's then metabolized again by something called aldehyde dehydrogenase, which is ALDH, and that converts it into an acetate, and or ethanoate. It then is converted into water and CO2. But then also some of it binds to coenzyme A and forms acetyl coenzyme A, which is involved in different metabolic pathways. This is the primary method of metabolism, but alcohol is also metabolized or can be metabolized by a cytochrome P oxidase enzyme called CYP2E1. This is the preferential pathway. But the thing is, with alcohol, you have to ingest such a large amount, relatively speaking, to obtain recreational effects, that the ADH, the alcohol dehydrogenase, is quickly saturated because alcohol has a very high affinity for it, and alcohol has a very high concentration in recreational use. So this is quickly saturated, and in very high amounts, the body primarily relies on CYP2E1. One interesting thing right off the bat is that through this ADH, ALDH pathway, the fact that the ADH gets saturated so quickly results in the elimination of alcohol, that is, the elimination of the drug from the body. Instead of following first-rate kinetics like most drugs, following an exponential decrease in concentration, it follows zero-order kinetics, and instead of an exponential decrease with half-lives, it has a constant rate of elimination. So, like if this is the rate of elimination where this axis right here is concentration, and this one here is time. A drug following first-order kinetics would have an exponential decay, something like that. That's probably not a good illustration, but with alcohol, in normal amounts, like not extremely high amounts, it'll follow zero-order kinetics and have a constant rate of elimination that is a straight constant rate. So I thought that was interesting to mention because, most drugs follow first-order kinetics rather than zero-order. But then another interesting thing to mention is that when you take a large amount of alcohol, when CYP2E1 gets involved, it actually changes and then follows first-order kinetics based on poorly understood mechanisms. It's thought that CYP2E1 is the primary mediator, but also there's unknown mechanisms that are thought to contribute to the conversion of the constant rate elimination to exponential elimination at very high doses. Here's a little side note. I just found this interesting and worth mentioning. So I wanted to go a little bit into other... Fucking hell. So I wanted to get a little bit into other alcohols. With ethanol, you have something looking like that. And with methanol, you have just one carbon. So COH. But methanol is interesting because it follows the same ADH, ALDH pathway as ethanol. You start off with MEOH, methanol, and it is metabolized by alcohol dehydrogenase, unsurprisingly. And what it forms is, instead of acetaldehyde, it forms formaldehyde. And formaldehyde is a lot more problematic in terms of toxicity because formaldehyde then goes through aldehyde dehydrogenase to form... There's fucking water on my board. Formic acid. Um, which is extremely damaging to the retina of the eyes, and even a small amount can lead to permanent blindness. So something I found quite interesting is that the actual antidote for methanol poisoning is ethanol, because when you drink methanol, or well, if someone drank methanol, I would hope you wouldn't want to drink it. It's going to be competing for these alcohol dehydrogenase enzymes, and when you drink ethanol with that, the ethanol will also be competing. So methanol by itself isn't very toxic, compared to formic acid and formaldehyde. So if you have enough ethanol saturating those dehydrogenase enzymes, less of the methanol will be converted to formaldehyde. The alcohol will also be, or rather the acetaldehyde produced by the conversion will also be competing for the ALDH. So you're going to have less formaldehyde, less formic acid forming if you ingest alcohol after ingesting methanol, or rather ethanol after ingesting methanol. And the same is true for ethylene glycol poisoning. Ethylene glycol is a diol. That is, it has two hydroxyl groups attached by an ethyl group. And it's also rather toxic, more so than ethanol, and it follows the same general pathway, even though it's a diol. It has two groups, but it still is processed by ADH and ALDH, and so the antidote for ethylene glycol poisoning is also alcohol. I just thought that was interesting to mention, because people rarely think of alcohol as an antidote for anything, but if you have methanol poisoning, I mean, you're going to get drunk off your ass on ethanol, but you're not going to die, hopefully, if they administer it in time. So in terms of toxicity of other alcohols, you can loosely put them on a bit of a continuum here, with methanol being the most toxic of the alcohols, followed by isopropanol, which is about twice as toxic as ethanol, but not nearly as toxic as methanol. So then you have ethanol, and then after this, the trend is, well, isopropanol is larger than ethanol. It has more carbons in it, rather. But generally after isopropanol, anything longer tends to be less toxic. So, like if you add butanol, it's going to be less acutely toxic than ethanol. So if you have a tertiary alcohol, like for example, tertiary amyl alcohol, this carbon is bound to 3 other alkyl substituents. These tend to be a lot less toxic than primary alcohols and secondary alcohols. And then with primary alcohols, the longer the alkyl chain is, the less toxic it is, generally. This is generally speaking. I mean, it might be less acutely toxic, but there's also things like whether it's a carcinogen. With some of the longer chain alcohols like hexanol, cyclohexanol, you're going to see more long-term effects, of course, due to the ability for them to cause cancers. But acute toxicity just is a reflection of what it's going to do to you if you drink it in the next, whatever, 24 hours, 48 hours, etc. So that's most of the kinetics I wanted to go over. I guess there's one more thing I should probably mention. There are a lot of potential interactions with alcohol. I'm not really going to get too deep into interactions with alcohol here because there's so many. I mean, the simplified version is, alcohol is a depressant. If you take other depressants, it's going to potentiate them. The longer explanation would have to do more with its pharmacology and how it has such a diverse array of targets and how many depressants share those targets. So that's more for the dynamics section of this, but one kind of interesting thing I thought I mentioned is that it can cause so-called dose dumping, which is a phenomenon where if you ingest a solvent, I mean, even water is a solvent, but in this case, the solvent would be ethanol. If you ingest a solvent after ingesting a time-release formulation of a drug, like for example, if you have like Adderall XR extended release. I mean, I don't know if this is specifically prevalent to Adderall, but something like that. It can cause if the coating on the drug that causes it to be extended release is soluble in ethanol, then the ethanol that you consume after taking that dose can dissolve the extended release coating and then cause it to so-called dose dump, all release at once. So I thought that was worth mentioning because, even if it's not a drug that you would think would have interactions, it could cause something like that to happen as well. And, there are other drugs that can affect the alcohol dehydrogenase enzymes and the CYP2E1 enzymes. And those drugs, of course, may have interactions with alcohol as well. So that's about it in terms of kinetics. So on to dynamics. So in general, the dynamics of alcohol are still poorly understood compared to other drugs, especially for such a drug, or rather for a drug that has such high use, such high prevalence in society. And the reason that isn't because of a lack of research. It's because of the biochemical properties of ethanol. It's a very small molecule, and you have to take a lot of it to get a recreational effect. Some drugs might have like an affinity of like 1 nanomolar. That's very high affinity. Alcohol has to be present in very, very high concentrations to achieve its effects. Even like 20 micromolar. And a micromolar is 1,000 times more than nanomolar. So that makes it difficult to use traditional methods of ligand binding assays. And so you have to rely on functional assays, which kind of opens its own can of worms. But, in recent years, I guess within the last decade, or rather maybe 2 decades, through functional assays, it's been determined a number of sites that alcohol seems to have an effect at. The exact quantification of that effect is hard to determine, but we know that it acts there, and we know relatively, we know about how it works there. So this is all kind of a bit iffy, but there is a lot of evidence supporting these sites. So the primary site that people kind of jump to when they think of alcohol pharmacodynamics is the GABA A receptor, and it has been shown to have activity at the GABA A receptor. It's theorized that its primary effects are through the GABA A receptor, but also it has many different targets, and it has a very diverse profile, and all of those matter in terms of its effects. So it's not like GABA A is the only thing that determines the effects of alcohol. So I'm just going to list some of these out. So we got, and then discuss them. So we got GABA A. So I'm going to write the activity that it performs, or is thought to perform, on the right. So it's a positive allosteric modulator of GABA A. I'll get more into details later. But then we got N-methyl D-aspartate receptor, NAM, Vietnam, negative allosteric modulator. All right, so we also got the opioid receptors, everyone's favorite. And there it acts as a positive allosteric modulator. At the serotonin receptors in general, as well as the ionotropic 5-HT3 receptors, which are serotonin receptors, but generally are, they're different in the effects of most other serotonin receptors. It acts as a positive allosteric modulator at both of those types, rather at both the metabotropic G protein mediated serotonin receptors and the ionotropic serotonin receptors. So what else? We got the AMPA receptors. Oops. Where it acts as a negative allosteric modulator. You're probably noticing a trend now. You know, it's very modulatory in action. It doesn't have many direct, or it doesn't seem to have many direct agonist or antagonistic effects at, you know, an orthosteric site. It tends to affect the allosteric site and act in a modulatory fashion. So next we have the kainate receptors, where it is a negative allosteric modulator. Oop. Lost my light, and now you hear a train. Ain't that something? So the acetylcholine receptors, we got the muscarinic as well as the nicotinic. Acetylcholine. I always forget which ones are capitalized here. Receptors, and at both of those it acts as a positive allosteric modulator. We have the glycine receptor. It is also a PAM. So moving on to other targets, alcohol acts as a glycine reuptake inhibitor, or GRI. That is, it inhibits the reuptake of glycine back into the presynaptic vesicles after release, and prolongs the time that glycine has clearance in the synaptic cleft. And also it acts as a adenosine reuptake inhibitor, which is a bit interesting to me. So an ADORI, ADORI, adenosine reuptake inhibitor. Same thing, but, you know, with adenosine rather than glycine. And then next we have its activity at certain ion channels. So at the L-type VDCCs, or voltage-dependent calcium channels, it acts as a, let me just specify, reuptake inhibitor. That's not an I. So at the VDCCs, or rather the L-type specific VDCCs, it acts as a blocker. It, through some mechanism, probably through, you know, a site within the pore, causes the channels to not allow the influx of, in this case, calcium after voltage-dependent activation. And then finally we have the GIRK. That is GIRK. That stands for G protein coupled inward rectifying potassium channels, where it acts as an opener. All right, so that's a lot of targets, right? And this isn't necessarily all of its targets. This is the ones that have been elucidated through functional assays. So I'm just going to briefly go over each one. The activity at each receptor kind of has implications for its overall effects. And so, you know, I'll just start off from the top. So GABA A, primary inhibitory receptor in the brain. It's an ionotropic receptor that allows influx of chloride into a cell. So a positive allosteric modulator of GABA A would increase the clearance of chloride into the cell, therefore increasing polarization of the neuron and reducing the chances it'll fire. So it's inhibitory. The neuron it's associated with is inhibited by activation. And positive allosteric modulator means that, just to briefly go over PAM versus NAM. So a positive allosteric modulator will either increase the affinity of an orthosteric ligand for a site, or increase the biological response that the binding of an orthosteric agonist elicits. So what that means is it's a modulatory molecule. It binds to a separate site than the orthosteric site. It binds to an allosteric site, and it either increases that receptor's ability for orthosteric ligands to bind to it by creating some conformational change that maybe exposes the binding site in a way that's more favorable for orthosteric binding, or when an orthosteric ligand binds to it while the allosteric ligand is also bound, it somehow increases the response that is elicited, the biological response. So the channel pore might open for longer, the channel pore might open more efficiently, whatever, So, and then a negative allosteric modulator, or NAM, essentially just does the opposite. It reduces the efficiency, the affinity, and the duration, and or any of those. Sometimes both, sometimes just one or two of those aspects. So moving on to the NMDA receptors, the N-methyl deaspartate receptors. I covered these in a video about NMDA receptors and their antagonists specifically, but there it acts as a negative allosteric modulator. And NMDA receptors, like I said in my other video, they're like the excitatory cousins of the GABA receptors. That is, when you agonize an NMDA receptor. Sorry, I got a cigarette in my mouth right now. So when you agonize an NMDA receptor, it helps facilitate the generation of an action potential in the associated neuron. So there it acts as a negative allosteric modulator. So there's a bit of a trend here. It positively modulates the GABA A receptors, which creates an inhibitory response. It negatively modulates the NMDA receptors, which also causes an inhibitory response. Okay, so opioid receptors. Positive allosteric modulator. That means that endogenous opioid peptides, hormones like the endorphins, it increases their activity at the opioid receptors by binding to an allosteric site. So you're going to have increased endorphin signaling, I guess. Serotonin receptors, including the 5-HT3 receptors, as well as many other metabotropic serotonin receptors. Positive allosteric modulator. I'm assuming this probably plays into its effect on mood, inhibition, whatever. I don't know exactly. There are many roles that serotonin receptors play. So, I mean, like, that could mean a lot of things. You have the AMPA receptors, negative allosteric modulator. I mean, AMPA receptors also help modulate the excitation of neurons. Same as kainate receptors and also glycine receptors. So AMPA receptors, kainate receptors, and glycine receptors in different ways affect the excitatory states of neurons. In terms of the glycine receptor, it works a bit different than the kainate and AMPA receptors. And these all work hand in hand with NMDA receptors. Usually, for example, a kainate receptor might introduce the initial catalytic influx of a positively charged ion into the cell, which then NMDA receptors respond to. The influx of the positively charged ion causes the efflux of the magnesium blockade on the NMDA receptors, which then act as sort of a mass influx propagator. I don't know. I mean, they respond to this slow increase of positively charged ions with a massive explosive increase. And there's sort of a chain reaction. So the kainate receptors and the AMPA receptors, they both work with the NMDA receptors indirectly to facilitate neuronal excitation. So negative allosteric modulation of both of these will affect NMDA-associated neurons and reduce their capacity to form an action potential, the same as the NMDA receptors. The acetylcholine receptors, we have the muscarinic and the nicotinic acetylcholine receptors, where it both acts as a positive allosteric modulator. So it's procholinergic, it's not anticholinergic. Drugs like diphenhydramine or other deliriants usually act as negative allosteric modulators or antagonists at the acetylcholine, or specifically the muscarinic acetylcholine receptor. So we have positive allosteric modulation there. That's interesting. I'm not really sure exactly what that means in terms of implications of and effects, but I find that interesting, and I like to see if I can find out more information on that. So glycine reuptake inhibitor, like I already kind of said, glycine in the synaptic cleft is brought back to the presynaptic vesicles by way of transporter proteins. In the case of glycine, they'd be glycine transporters. And alcohol inhibits that by binding to the transporters and preventing glycine binding to them or otherwise disrupting their function. So that means that glycine will stay in the synaptic cleft longer. And also it acts as a positive allosteric modulator of glycine, so that this potentiates that as well. And then we have adenosine reuptake inhibitor. Now, adenosine plays a lot of roles in sleep. I think caffeine affects adenosine receptors. Interesting that it inhibits reuptake. My gut reaction is that that would make you sleepy, but I don't know. Not sure. That's speculation there. So, L-type voltage-dependent calcium channel blocker. Alcohol has this trend of modulating a lot of different ion channels. The GABA, NMDA, HT3. A lot of these are ionotropic receptors, and VDCCs are also ionotropic, or rather, they're voltage dependent ion channels. So the voltage dependent part means that the inside of the neuron, the postsynaptic neuron, needs to reach a certain state of electrical potential, rather membrane potential, for these receptors to then activate. That's why they're called voltage dependent or voltage gated, VDCC or VGCC. And these ones specifically are ones that allow calcium into the cell or neuron. So by blocking that, calcium generally speaking, being a positive ion, will help depolarize the membrane potential and increase the chance that an action potential will form and cause the neuron to fire. So when you have something blocking these VDCCs, calcium influx is dampened, and it reduces the chance of the neuron firing. So inhibitory, much like a lot of these other ones. So the GERK opener, these are G protein coupled inward rectifying potassium channels. They often are adjacent to G protein coupled receptors, like the GABA B receptors, and function through G protein signaling to open and allow the influx of potassium ions, which, depending on the context, can help stabilize the neuron. So yeah, GERKs regulate potassium influx, which helps stabilize the membrane potential. Okay, so now that we got that out of the way, onto the reward pathway. So acetaldehyde, the metabolite of alcohol created through alcohol dehydrogenase, it plays a vital role in reward pathway activation. I'm talking about the mesolimbic reward pathway. When you use alcohol acutely, that is, you're not using it every day, you're not using it it's not sustained chronic use. It causes dopamine release in the mesolimbic pathway, which activates, of course, the dopamine 1 receptors in particular. And what that does is causes the activation of intracellular protein kinase A. And pKa is a ubiquitous enzyme found in cells that's often used to phosphorylate targets by acting as a secondary messenger, essentially. It can affect many targets, but in a lot of cases, it affects transcriptional targets. That is, targets that affect transcription of genes. So it'll affect the epigenetics of the cell in question. That is the way that the genes are expressed. This isn't a huge issue when you just take it like once in a while, but if you take alcohol every day, you're going to downregulate those dopamine receptors because you're going to have chronic pKa activation, which in turn is going to phosphorylate the CREB protein, which stands for CAMP, cyclic adenosine monophosphate response element binding protein. And when you phosphorylate that, it affects gene expression. So if you have overstimulation of these dopamine receptors, it's going to cause pKa activation, and then that's going to cause CREB phosphorylation, which is going to then in turn affect gene transcription, particularly in this case, it'll reduce the expression of the dopamine 1 receptors because they're being overstimulated. So that's how the cell responds to that. Now, also in chronic use, it'll affect CREB itself and NMDA receptor function as well. And it does so through MAPK and ERK, as well as CAMK pathways or mediated pathways. And that's kind of a lot, so let me just go over that. So it affects these targets by these two pathways. One is called MAPK/ERK, and what that stands for is MAPK is mitogen activated protein kinases, and ERK is extracellular signal regulated kinases. So these are both kinase pathways, protein kinases. These are enzymes, and activating these pathways causes these longer-term effects of alcohol. Then the CAMK pathway stands for calcium/calmodulin-dependent protein kinase, which also, like I said, it's one of the pathways that sustained use of alcohol will affect, or rather will elicit its effects in the brain through. And the way it does so is by inducing expression of delta phos-B, especially in the nucleus accumbens. Now, delta phos-B is a very interesting protein to me. It's implicated in almost every type of addiction, and it's a protein splice variation of phos-B. So phos-B, it's also called G0/G1 switch regulatory protein 3. Bit of a mouthful, but what phos-B does is when it's overexpressed due to the MAPK/ERK and the CAMK pathways due to CREB phosphorylation due to protein kinase A activation, sustained activation. What it does is it causes delta phos-B to be overexpressed, and that has been implicated with addiction-related neuroplasticity changes. That is, changes in neuroplasticity that are indicative of addictive behavior. Neuroplasticity isn't always a good thing. I mean, you don't want your brain to be plastic in the regions where addictive behavior, compulsive use are cemented, I guess, to oversimplify it. But so I find that really interesting. And this explains why, for months after you quit something, you'll still have cravings for it. You'll still have compulsions to use it. And that's because delta phos-B isoforms have a very long half-life. And so you have these symptoms for months, even after you cease usage, because they're still present in the nucleus accumbens. They're still overexpressed, and it takes time for them to return to a baseline level. I think that about kind of covers most of it. I mean, there's other aspects that I could get into, like toxicity. They can kind of be summed up by, alcohol can cause liver, kidney, etc., cancer. It's a type 1 carcinogen. I think most people know that, alcohol can cause cancer, as well as, organ damage and detrimental effects in functioning, like, liver damage and all that. So I'm not really going to get into that. I find the pharmacokinetics and dynamics more interesting than the toxicology personally. So that's really what I wanted to discuss. I think I went over it pretty thoroughly, I guess. There's some things I probably could have gotten into more detail with, and some things that I probably didn't bring up that, are relevant here. Yeah, let me just close this video out. Yep, so this is an outro. Welcome to the outro. I think I covered most of what I wanted to say there. But yeah, shout out to Cruel Worm, the homie on Discord, for suggesting I do this video. I hope that it wasn't too tangential. I know I didn't really in some of my other videos, I really go deep into trying to explain everything in a way that I hope that is digestible. I know in this one I kind of went fast and I didn't really explain certain things in depth like I would usually, but if there's anything that anyone wants cleared up, feel free to comment if anyone watches this. And also you can hit me up on Discord. My username is Shulgs, like my channel name, and I'm happy to discuss pharmacology with whoever, whenever. I don't care how much you know, how much you don't know. I just love talking about it. And yeah, I think that's about it. Yeah, it was fun to talk about. I'll catch you guys later.